Alrighty, I'm going to go ahead and get started. Welcome, everybody. My name is Owen Snedden. I am the president of the College of Natural Sciences and Mathematics Student Council here at Cal State Long Beach. I just wanted to welcome everybody to the Crafoord Lecture event with Dr. Andrew Knoll. Going to continue introductions by passing the microphone to Dr. Curtis Bennett, Dean of the CNSM.
Thanks, Owen. I'm Curtis Bennett, Dean of the College of Natural Sciences and Mathematics, and I'd like to welcome you all here today. The lecture that we give, the prize lectures, is a long-standing event from the CNSM Student Council. It is run by the students, for the students and faculty, and it's one of the great things that they do for all of us. I don't want to say a whole lot more about that. We're very, very happy today to have Dr. Andrew Knoll. But to introduce Dr. Knoll, I am now introducing Dr. Nate Onderdonk, the acting chair of the Department of Geological Sciences, soon to be the Department of Earth Science. Dr. Onderdonk.
Thank you. So thanks everybody for joining us today. It's my pleasure and honor to introduce and welcome Dr. Andrew Knoll from Harvard University, who will be talking today about some of his research on the earliest history of life on the planet. And I think this is a fascinating topic because if you think about the amount of information that these scientists are able to squeeze out of just the very few rocks that are actually preserved on the planet from hundreds of millions of billions of years ago, it's pretty impressive. So Dr. Knoll started his academic career at Lehigh University, where he got a bachelor's degree in geology and then went on to Harvard for his graduate degrees. He then taught for a little while at Oberlin College before joining the faculty at Harvard University, where he's been since then. He's served in both the geology department as well as the biology department. Last year, he was awarded the Crafoord Prize for Geosciences. And just a few words about that prize. This is given out by the Royal Swedish Academy of Sciences. It was established in the early 80s for several categories that are not already covered by the Nobel Prize. So those categories are geosciences, biology, astronomy and physics, as well as a rheumatoid arthritis category. So they're given in alternating years. And with that, I will turn to Dr. Knoll and thank you for coming.
Okay, well, thank you, Nate. Let me just share my screen. Hopefully this will work. Okay. Good, I hope everybody can see the screen. Let me first say thank you to Nate for the kind introduction, and thank you to Owen and Natalia for inviting me to be with you today. I'm sorry I can't be with you in person, but I'm delighted to spend the day virtually interacting with people in Long Beach. My title is, in fact, The Deep History of Life, but what I hope that I can impart to you today is that it's actually most of the history of life. And so the question is, how can we reconstruct life's long history? How can we reconstruct the environmental context in which evolution has played out through time? And in general, how can we understand this conversation between Earth and life that has been going on for most of our planet's history? So let's start at a place that everyone will be familiar with. I think if you know anything about the fossil record, you know that long before there were humans, there were dinosaurs, mighty monsters that were around for about 150 million years or more. If you know a little bit more about the fossil record, you might know that at the time when dinosaurs were the predominant members of land communities, there was a group, another extinct group, belonging to the cephalopods, the group that contains squid and octopus in the seas. You see that fossil to the right, that's called an ammonite. And those were some of the most diverse animals in the oceans at the time of the dinosaurs, some of the major predators and scavengers in the oceans, roles today played by fish. And if you know even a little bit more about the fossil record, you'll know that long before there were dinosaurs and ammonites, there were animals, including the things you see at the bottom here, trilobites, which are early arthropods, the group that contains insects, lobsters, shrimps, etc. And these were some of the most diverse organisms on the seafloor between about 520 and 250 million years ago. Now, this next slide tries to put that record in several types of perspective. I really like this diagram in the upper left. It comes from some work by Ron Milo's group at the Weizmann Institute in Israel. And a few years ago, they made an estimate of just the biomass, that is the weight of all the different kinds of organisms living on Earth today. And if you look at that, the left-hand side there, you'll see that in their estimate, most of Earth's biomass consists of plants. Bacteria are second, then you have fungi, archaea, which is another group of simple organisms organized at least broadly like bacteria, things like protozoa and algae, and that little red rectangle in the upper right is animals. Animals are only a very minor percentage of all the massive organisms living on this planet. Now, it has to be said that in a sense, plants cheat, because if you go out and measure the biomass of a tree outside your building, you'll find that something like 90% of its tissues are not living. It's things like wood, which don't really become functional until the cells have died. And if you only take the living biomass, the living cells of plants, then it turns out that most of their biomass is bacteria. So bacteria kind of rule the roost. Animals from this metric are rather minor. Now, if we drop down to the lower left, what you're looking at there is something that's called a universal phylogeny. It's an attempt to document and display the evolutionary relationships among all organisms living today, really based on comparisons of sequences for DNA and sometimes proteins. And there are two things to point out here. First is that if you look at the major architecture of that tree, there's three great branches. One of them is the eukaryotes. That's organisms like all of us that have membrane-bounded nuclei and that sort of thing. A second is bacteria, simple organisms, pretty much as advertised. And then only in 1977 was it shown that among those simple microorganisms that live in various environments on Earth, there was a whole nother branch of the tree called archaea, which is still only now coming to light. And so there are a lot of things to argue about in terms of the relationships shown in that tree, but the idea that there are three major branches holds. By the way, one major group of archaea was only described for the first time about 8 years ago, and it turns out that probably contains the sister groups or closest relatives of eukaryotes. So it's very likely that at least a good portion of eukaryotic biology originates within the archaea rather than being a sister to them. But the other thing to notice here is that the animals, all of the animals we see today, all the animals found in the fossil record reside on one small distal branch of the tree. That suggests two things. One is that there's a lot more diversity out there. And two is most of that diversity is microbial. So to the extent that we can look at that tree of life as a timeline, it suggests that long before there were any animals, there was life, largely microbial life. And then if we take that thought and look at just a timeline of Earth history, what this shows is that the oldest evidence we have in the fossil record for animals is in rocks that are about 575 million years ago, or And yet the Earth itself is more than 4.5 billion years old. So the question that I want to address this morning is, what happened back in this area that we're shown with question marks here? Phylogenies lead us to predict that there was a diversity of microbial life, but the question then is a practical one. Can we actually demonstrate that history and come to understand it in a historical sense? Now, I thought I'd just take a slide to talk about how I got into this business in the first place, because I understand that there's a lot of students who are probably listening to this, and you're all somehow engaged in trying to understand the path that you want to take on your own journey. So I will say without equivocation that when I was 18 years old and headed off to college, I hadn't the slightest idea what I wanted to do in life. I came from a rural upbringing. I knew basically of three professions: doctors, lawyers, and engineers. I knew I didn't want to be a doctor or a lawyer. I was pretty good at math, so I went to engineering school. And then by the end of my first year, I knew I didn't want to be an engineer either. So there I was entering my sophomore year in college, really with no idea what I wanted to do. And so really just out of almost desperation, I took a whole bunch of science and math courses. And I found that I really loved a course in earth science and geology. And I also took a biology course that I really loved. And in maybe the only real insight I ever had in my life, I was sitting in my dorm room one night, and I realized that maybe geology and biology were not the separate universes that they seemed to be from the way they were taught at that time. Maybe if I understood something about the Earth, that would help me to understand the biological record. And maybe if I understood the biological record, that would help me to understand Earth's environmental history. And that thought just struck me, and it really set me on the course that I've followed to this day. The other important thing that happened when I was an undergraduate was I was taking a botany course. We had to do a term paper, and I ended up doing my term paper on an idea that was very exciting and controversial at the time, which is the idea that in the eukaryotic cell, the major organelles that are responsible for energy metabolism, the mitochondrion, and in photosynthetic organisms, the chloroplast, were actually derived by once free-living bacteria that were basically gobbled up by eukaryotic hosts and reduced to metabolic slavery. That idea came from the work of Lynn Margulis, who you see here. And as I was doing my term paper, I was learning about the work of a man named Elzo Barghorn at Harvard, who was actually discovering a record of life that was much older than that of animals. And so that got me excited. Fortunately, I was accepted to work with Elzo as a graduate student, and that's how I got interested in the early record of life and environments. Just as a coda of that, I have to say that my first year in graduate school, I was sitting at my desk, and Elzo brought this woman over to introduce me. He says, "Andy, you should meet Lynn Margulis." And to me, that was kind of like, I don't know, meeting LeBron James or something like this, because she had become my hero just from reading her work. And Lynn, for the next 40 years until her death, was a very kind and supportive friend. So I'm always grateful for that. Well, let's get back to science. One of the early places I had an opportunity to work was the island of Spitsbergen, which you see here. Now, this is Spitsbergen in the middle of summer, so in some ways it's not the most promising place to spend your summer. It's about halfway between the northern tip of Norway and the North Pole. You get around basically by flying in a helicopter or being in a boat, and then once you're let off, you use snowshoes and skis to get around. But there's a reward for this kind of work. There are beautiful outcroppings and exposures of rocks. The ones that you're looking at are about 800 to 720 million years old. That is, they're significantly older than any rocks that contain evidence of animal life. And so the question is, might these contain a record of this earlier life implied by phylogenies? And just to give you a sense of just how good the outcroppings are here, if we put the Empire State Building at the base of this cliff, its radio tower would just peer over the top. So it's an extraordinary geologic record. So the question is, how are these rocks going to help us to move the evidence of life more deeply into the past? Well, it turns out there are lots of clues to both life and environment that have been encrypted into these rocks. So in the upper right, what you see are some carbonate rocks like limestones today that were laid down on the order of 800 million years ago. And the first thing you notice is that there's these kind of wavy laminations. If you look for wavy laminations like that forming today, it tends to happen on places like the Bahama Banks, where you have microbial communities that cover the shallow seafloor, and those interact with incoming, in this case, lime muds. So right away, to a person with a geological eye, that implies that there were microbial ecosystems in this area. The other thing you see in that upper right image is there's like a bowed upward area in the upper part. That's what geologists actually call a teepee structure. And those form in the supratidal zone, that is a zone that's commonly left high and dry during the tidal cycle. And when that happens, you actually get precipitation of calcium carbonate cements within the sediment, and the pressure from that causes the beds to buckle. So what we learn here is that simply looking at the physical features of that piece of rock, we learn something about life and something about environment. Now, if I take a piece of that carbonate rock, cut a paper-thin slice out of it and look at it under a microscope, all I'll see is interlocking carbonate crystals. But if you notice, there are these sort of black pancake-shaped structures within that rock. Those are nodules of chert, that's fine-grained quartz, or SiO2, and those nodules form within the sediment soon after they formed. And it turns out that chert acts kind of like bioplastic in that it preserves over long intervals of time morphological and chemical features of the sediments within which it formed. So if I take a paper-thin slice of that black stuff and put it under the microscope, what you see is what this image shows in the lower part of the diagram. These are cells, 800 million-year-old cells, only a couple of microns in diameter. We think these are cyanobacteria, which are photosynthetic bacteria, still common today. So now we've learned something interesting, that not only are there textural features of the rocks that suggest the presence of biology, but they're actually microbial fossils that are preserved in these rocks. Just to give you one example of just what we can do as far as interpreting them when the conditions are good, up in the upper left here, you see this interesting fossil. It's about 30 microns across. There was once a single cell in the middle of that structure. That's basically degraded into that sort of dark goo. But the cell during life was surrounded by numerous extracellular envelopes or sheaths. And they were elongated in the downward direction because these actually formed a stalk that kept the cell right at the sediment water interface. And we find them as individuals, we find them as just crusts that formed on the seafloor. And we can know enough about them by looking at populations that we can reconstruct their life cycle, like you see on the bottom. So, we can say a lot then about these, what they were like, what their life cycle was like, where they lived. And if we go to places on the modern world where we find environments similar to those that we've inferred from those rocks, this is the Bahamas, you find in the intertidal zone these black crusts. And those black crusts, if you look in the lower left, are made by single-celled cyanobacteria that make these sheaths that form stalks, just like the 800 million year old fossils. So we have a very close comparison among morphology, life cycle, and environment, really across 700 to 800 million years. And it turns out that when you look in different types of environments represented by rocks in this succession, there's actually quite a diversity of life. On the right are a bunch of things that we think are all cyanobacteria, these photosynthetic bacteria. The ones on the left are all fossils of eukaryotic organisms. The upper center, for example, it's about 100 microns long. That vase-shaped structure was made by something called a testate amoebon, a group that we can study today because it still is with us. On the left is actually a multicellular fossil that is very similar to some living green algae. In the lower center, there's again something that's about 0.5 millimeter in diameter, so it's clearly eukaryotic, a cyst-forming eukaryote, although we don't know really what kind of organisms made them. So the first lesson is there is actually a fossil record of life before animals. Now, it turns out the most conspicuous fossils or most conspicuous evidence of biology we find in these rocks are actually structures called stromatolites. And these are really microbial reefs. So if you look on the right-hand side, you can see that there are these structures that were nucleated on cobbles. And then we have these laminations sort of bowing upward that accrete through time. Those, as it turns out, are made by microbial communities that trap and bind sediments and then become mineralized. And we know how those work because if you look in the lower left, there are still a couple places on Earth today where cyanobacterial communities make these stromatolitic structures. This is a place called Shark Bay in Western Australia, which is kind of the mecca for this kind of study. And if you look in the upper left, back in Spitsbergen, right in the middle of that cliff behind me there are these sort of lozenge-shaped beige bodies. They're about 4 or 5 meters thick, and those are patch reefs. We're used to thinking that reefs are made by corals, but for most of Earth history, reefs were made by microbial communities. And so stromatolites show us that microbial communities covered the seafloor pretty much all down to the bottom edge of the photic zone. And then there's chemical evidence for life. It's interesting that I always tell my students that when you die, the last part of you that will be available for future generations to contemplate will be your cholesterol. That's something to keep in mind at lunch. It turns out that sterols and some other kinds of molecules actually preserve reasonably well in the geologic record. DNA doesn't preserve well. The oldest fossil DNA that we know of is only about 2 million years old. Proteins don't preserve very well because, like DNA, they're good food for microbes, but sterols, different kinds of lipids, preserve pretty well. So we find on the upper left there, that's a sterol structure. We find the geologic remnants of sterols called steranes, and they tell us about different groups of eukaryotic organisms that are not well represented by body fossils. And then there are structures like the ones below that called hopanes, which also tell us about bacterial diversity. And then even more generally, individual microbes are small, but in the aggregate, they can actually influence the chemical composition of seawater. And so one of the things that we've been looking at ever since I was young is the carbon isotopic composition of carbonate rocks and organic matter found within them. Turns out that there are 2 stable isotopes of carbon. About 99% of all carbon on Earth has 6 protons and 6 neutrons, called carbon-12. About 1% has an extra neutron, it's called carbon-13. Also in the parts per trillion, there is carbon-14, which is a radioactive isotope that breaks down to nitrogen on scales of thousands of years. The reason I bring it up is that when photosynthetic organisms take carbon dioxide and fix it into organic matter, they actually discriminate between carbon dioxide that has carbon-12 and carbon dioxide that has carbon-13. So if you look today in a marine environment where there are carbonates, the carbonates tend to have values, around 0 per mil on this arbitrary scale, and the organic matter is depleted in carbon-13 by about 25 parts per thousand, something that's easily measured using mass spectrometers. And if you look in Spitsbergen, there is about a 28 parts per thousand difference between limestones and the organic matter found inside them. That is really evidence that 800 million years ago, there was a biological carbon cycle. I could do the same story for sulfur and nitrogen isotopes, and it's pretty clear that there were microbial sulfur and nitrogen cycles as well. Okay, so what you're looking at here is some modern microbial mats we've been working on in the Turks and Caicos Islands in the Caribbean, because that actually shows us how the geologic record forms and allows us to understand what does and doesn't get preserved in the record. And as we've seen, we have, in these rocks that are 800 million years old, the fingerprint of life is everywhere, in microfossils, in stromatolites, and in chemistry. So let's go back in time. Let's double the age. And here's another place I worked on in the past, something called the Bilyach group in northern Siberia. And again, what you're looking at here in this cliff are carbonate rocks laid down now about 1,500 million years ago. But again, when we look at them, there are microfossils of cyanobacteria, which you see that filamentous structure. These are 10 micron scales. There are eukaryotic organisms as well. I really like this picture in the lower left for 2 reasons. One is that my colleague here in the field, Misha Semikhatov, Misha was precisely 2 meters tall when he had his hat on, so he was just a wonderful person to use as a marker for your pictures. And you might say, well, I see why you took this picture. Right in the middle, there's this sort of round donut-shaped structure. That's probably a microbial reef. And you know what? You're right. So give yourself 5 points. But this turns out to be a 15-point question, because if you look at the surface that Misha is standing on, it's sort of rounded and curved. The little patch reef in the middle, might be the size of a small room, maybe a restroom, something like this. Misha's standing on a microbial reef that was larger than a lecture hall at Long Beach. And if you look in back, you can just see those vertical structures. He is actually standing in front of a wall of a stromatolite reef that would be larger than whatever building you're in now. So again, 1,500 million years ago, we had microbial structures making reefs of the dimensions of large coral reefs like the Great Barrier Reef today. Also, we can look at carbon and sulfur isotopes. We can look at molecular biomarkers. Again, 1,500 million years ago, the microbial world was thriving. So let's double the age again and go back to some of the oldest rocks we can look at and hope to get some chance of finding evidence of life. And that's something called the Warrawoona group in Western Australia. It's nearly 3.5 billion years old. This just shows you the fun you have when you're trying to get to see it. But what do we actually find? Well, The first thing we find is carbonate rocks are not common in this succession, but when you see them, they have stromatolites. I hope you can see the kind of cone-shaped structures in the center. Again, there are microbial ecosystems on the seafloor. These rocks have been heated or metamorphosed, if you will, so that we don't find molecular fossils like we did in younger rocks. But if you look at the carbon isotopes, carbonate rocks are around 0. Organic matter associated with them is around minus 25 to 30. There was a microbial carbon cycle at this time. There was also a microbial sulfur cycle. And then people have described microfossils from these rocks for a long time. And that's a little bit controversial, because what you see here on the left are some 10 micron spheroids made of organic matter from these rocks. And they look like they might be microfossils, but one of the things we've found is that when we look in younger rocks where we have a better idea what's going on, and specifically look where there's been hydrothermal fluid flow, we find that the hydrothermal fluids actually remobilize organic matter and precipitate it out as 10 to 20 micron organic structures. So the things you see here in the upper right, they're 10 to 20 micron organic spheroids, but they're not microfossils. They're structures made by hydrothermal fluid flow. And pretty much every rock we can look at deposited 3.5 billion years ago has been affected by fluid flow. So it's not clear where we stand with respect to actual microfossils at this time. So what do we learn then from this quick Cook's tour? Well, the available evidence says that life has been part of the Earth's system for most of our planet's history. You know, going back in time, we run out of rocks to look at before we run out of evidence for life. So life originated early in our planet's history and has been sustained for more than 3.5 billion years. Now, there's a lot of uncertainties about the nature of this early life, but one thing seems clear from the chemistry of the rocks in which we find this record, and that is that early life worked without oxygen. So what's interesting is that when we put on our more geological or geochemical hats, we find that basically for the first billion or more years of a geologic record of environmental history, we find that there was a world with no persistent oxygen gas in the atmosphere and no persistent oxygen in the oceans. What makes us think that? Well, there's several lines of evidence. First of all, as you see in the right-hand side here, these beautiful reddish rocks are something called banded iron formation. These kind of rocks, which are really the source of most of the iron that's been mined for the last 2,000 years, those kind of rocks are very common in the first billion years of Earth history and pretty rare thereafter. It turns out you can't make rocks like this today, because in order to make that rock, you have to be able to move iron through solution in the oceans, and that cannot happen if oxygen is present. So this tells us right away that we had an iron-rich, oxygen-poor ocean. There are also a number of minerals. For example, in the lower right, you're looking at a sandstone deposited about 2.7 billion years ago. And that rounded structure that you see, the gold in that, looked at under the microscope, is pyrite, iron sulfide. Turns out that even if there's a little bit of oxygen present, that will oxidize to sulfate on very short geologic timescales. So what this tells us is that that pyrite was weathered out of a rock that was exposed on the continent, carried down by rivers and deposited in an estuary without ever coming into contact with significant amounts of oxygen. There are other minerals, a uranium mineral called uraninite, siderite, which is iron carbonate, which tell the same story. Those kind of sediments are common until 2.4 to 2.2 billion years ago, and then you don't see them anymore. There's also some really interesting features of the sulfur isotopic record that just changes dramatically on this time scale. Before that, it's pretty clear that sulfur isotopes were imparted mostly by chemical reactions in the upper atmosphere that only take place when there's no oxygen around. After 2.2 billion years ago, we don't see that record anymore. So a number of lines of evidence tell us that more than 1 billion years into the history of life, we have a major change in environmental history, and that is that oxygen begins to accumulate. And what says our hero there is because there's only really one source of oxygen that is large enough to account for the oxygenation of the Earth's surface, and that's cyanobacterial photosynthesis. So it turns out, it looks like there is good reason to believe that cyanobacteria were able to make oxygen long before this great oxygen revolution, but didn't do so. And so people still argue about why this might be, but one thing that probably plays a role is that, for most of that first billion years of the geologic record, Earth increasingly seems like it was a water world. That is, there were islands that stuck out, maybe little bits of continental crust, but largely, if you had looked at the Earth from space 3.5 billion years ago, you would have pretty much seen water. Under those conditions, there's very limited weathering and erosion, which is what would generate phosphates, an important nutrient to go into the oceans. And also, because there was no oxygen, there was very little in the way of oxidants that can actually recycle organic matter once it's made. There wasn't much sulfate, there wasn't much nitrate. And so under these conditions, the amount of phosphorus that would be available for primary production would be rather small and might favor photosynthesis by kinds of bacteria that didn't use water as an electron source, didn't produce oxygen. And this little slide that you see here suggests that, almost no matter whether you think primary production was limited by electron donors or nutrients, that on the early Earth, rates of primary production were much smaller, maybe only a couple % of what they are today. It was a very different world. What happens then, we increasingly have evidence that around 2.5 billion years ago, we have the emergence of continents. This is thought to happen because basically the Earth's mantle is cooling, and so it can now support continents. Also, as water gets subducted by plate tectonics, it's more prone to stay there. So for whatever reason, the world starts looking more familiar 2.5 billion years ago. We see evidence in the geologic record for large continents, and that may have driven the changes that make cyanobacterial photosynthesis ecologically important and ends up changing the world. So how much oxygen was produced? The answer is not that much. And the reason we say not that much is we have a variety of different kinds of chemical signatures that we can look at to ask about the oxygen content of different water masses. And when we do that, almost universally, from about 2.2 to 600 million years ago, we have evidence of a little bit of oxygen in the atmosphere and surface ocean, but the deeper ocean remains oxygen-free. However, that was enough oxygen to support a new kind of life. Early on, life is bacterial and archaeal, but beginning, well, from the rock record about 2.7 billion years ago, we start seeing fossils that are confidently interpreted as eukaryotic organisms. We don't quite know what kind of eukaryotes, but we have both single cells that tell us actually that a lot of the features of eukaryotic cell biology were in place. We have, as you see in E and F there, simple multicellular organisms. Here you see some more of the multicellular organisms. And if you look on the right-hand side, the one in the middle is a simple multicellular filament about 1 billion years old that is a red alga. And to the right of that are some billion-year-old fossils that are green algae. So by about 1 billion years ago, we are seeing types of eukaryotic organisms that we actually can relate to living groups. What you're looking at here are the oldest known multicellular eukaryotic organisms found in some very well-dated rocks from China. This is work that I participated in, but led by Lanyan Miao, who's a very good postdoc in Nanjing. And, you know, if these fossils were found in rocks that were 300 million years old, they would be interpreted as green algae without any question. We don't necessarily know that they're green algae, but they are simple multicellular eukaryotes, probably photosynthetic. So very soon after we see evidence for eukaryotes in general, we see evidence of simple multicellularity. Interestingly, however, is that there'll be another 1 billion years before we see the complex multicellularity associated with animals and more complex algae and plants. So let's slide through that 1 billion years and look at this place, wonderful place, a World Heritage Site in eastern Newfoundland called Mistaken Point. It's about 565 million years old, and you can see that there's beautiful bedding surfaces preserved there. And in those rocks, we start to see complex macroscopic fossils. So what you're seeing here is a bedding plane, and basically the organisms that were living on that surface were buried in volcanic ash. This is kind of a geological or paleontological Pompeii. And you have a holdfast, you have a stipe, and then you have these structures at the top. Lots of these, a lot of diversity. And what are they actually trying to tell us? Well, what we think is that these organisms are early animals. They basically have upper and lower cell layers called epithelia, that all of the metabolism by these was done in these thin layers of metabolically active tissues. And then in the interior, they seem to have had either fluid or some kind of jelly, like the jelly we see in jellyfish today. They have no evidence of specialized organs for gathering and transporting oxygen. That is, there's no lungs or gills. So they exchanged gases by diffusion. There's no evidence of mouths and digestive systems. They fed by absorption of organic molecules and probably by what's called phagocytosis, that is individual cells capturing particulate organic matter. There's no evidence for muscular locomotion in these. And so these are very simple structures, didn't need much oxygen. And in fact, their size and dimensions would have been limited by the requirement that they gained all their oxygen by diffusion. So armed with that, where do those fossils fit on the tree of life? And what you're looking at in the upper left is a very simple tree, shows choanoflagellates, which are the most closely related protozoans to animals. And then you have a simple animal tree that has sponges. It has at the top the red ones, which are cnidarians, that's jellyfish, sea anemones, corals, and the like. And then most animal diversity is bilaterian animals with a bilateral symmetry, a front, back, top, bottom, left, and right. In between sponges and the groups in red is a group called placozoans, with only one really described species, this little thing called trichoplax that you see here. It's only a few millimeters long, and it's very simple. It contains upper and lower cell layers, a fluid-filled layer in between. It moves by means of cilia on lower on the lower surface of the cells, doesn't have muscles. It feeds by absorption and phagocytosis and exchanges gases by diffusion. In other words, except for its smaller size, the description of placozoans is very similar to the description of those fossils that we saw from Newfoundland. And I think most people, including me, would put the earliest fossil record right in here, between sponges and the diversification that would produce most of today's diversity. Now, within 10 million years of the time those rocks were laid down, we do start to see fossils of animals that are bilaterian. This thing called Kimberella does have a front, back, left, right, top, and bottom. You can see it has a mantle over some viscera. It has a muscular foot on the bottom, things that are today associated with mollusks. If you look in the upper right, it's a beautiful fossil from Arctic Russia, and you can see this sort of scratch marks in front of the body fossil. That suggests that this had something similar to what in mollusks is called a radula, which is really a scraping organ that helps it take in food. And then in the lower right, we actually find some of these fossils at the end of tracks and trails. So they were motile. And then within about 15 million years of that, we see the beginnings of the body plans that characterize modern diversity. In the beginning of the Cambrian period, we soon start to see arthropods. In the lower right, we see mollusks. In the lower left, we see annelid worms. And above that, we actually see vertebrate animals. So all of the basic phylum level body plans that we know of are becoming apparent in the Cambrian period. At the same time, as you see on the upper right, we see records of tracks, trails, and burrows that provide independent evidence of the growing anatomical and behavioral diversity of animals. And on that same time scale, we see evidence that Earth is moving out of this long intermediate period of low oxygen. And again, many different chemical indicators of environment tell us that oxygen is starting to increase. And by about 400 million years ago, then we have the oceans being pretty much fully oxygenated. What you're seeing here on the upper right uses the element cerium as it's found in limestones. And this is the work of Xianming Liu, who I've had the privilege of working with. And what she shows is that just when we start seeing these simple animals, we see evidence that the environment with oxygen is beginning to change. Now, how much oxygen? Again, to make the kind of animals you see in those 575 million, 565 million year old rock, the answer is not much. Some interesting work done by a guy named Dan Mills about a decade ago, in which they grew sponges at very low oxygen concentrations, and the sponges survived. Just a percent or a couple of percent of today's levels. Now, no one knows whether they could complete their reproductive cycle, but they could at least live. Now, it has to be said, now they concluded from that you don't need much oxygen to make animals. And I think for the various earliest animals, that's probably right, but the levels of oxygen in which they grew their sponges would actually kill most animals today. So we have to ask a more sophisticated question, if you will. Animals of different size, animals that have complex anatomy, locomotion, feeding type, and in particular, one of the things that we've found, and this is the work of my former postdoc, Eric Sperling, now at Stanford, carnivores need a lot of oxygen relative to other kinds of animals. In part, that's because they move around and catch prey, but also because, a lot of carnivores stuff a significant part of their body weight into their digestive system when they hunt, and that puts a very strong, if transient, oxygen demand on the organism. And what Eric found when he looked at, there's been a variety of ecological surveys of gradients, because in today's oceans, you can go to fully oxygenated to fully anoxic waters. And what Eric found looking particularly at polychaete worms, because they're so abundant and have different lifestyles, that in very low oxygen waters, you don't see much. But as you go to, 4 or 5% of today's oxygen levels at the surface, you start to see some. And by the time you're at about 10% of today's oxygen levels, you see a real diversity of individuals, as well as a real diversity of taxa. So simply going from 1 or 2% of oxygen levels to 10% would provide you with the environmental context for the radiation of animals that we see in these Ediacaran and Cambrian rocks. Here's another way of looking at it, also from work with Eric when he was a postdoc. And again, just looking at different features of the animal ecology with respect to oxygen, whether it's diversity or evenness, or for polychaetes, whether they're, the abundance and diversity of carnivores, the world really changes at around 8 to 10% of oxygen levels. There's good reason to believe that Earth went above that level on just the time scale when we see animals. So that then brings us to the last part of this presentation. There's one more change that happens on this same time scale, and that is, for the first time in Earth history, we see that eukaryotic algae are becoming major primary producers in the Earth's oceans. Before that, it was all cyanobacteria for more than 3 billion years. And again, I'm very proud that yet another of my former postdocs, Jochen Brox, has done this great work where he looked at rocks between 900 and 645 million years ago. He finds some fossil steranes, these sterol versions, or remnants of sterols produced by eukaryotic organisms, but nothing associated with photosynthetic organisms. Beginning around 600 million years ago, there's one place that has green algal steranes being abundant. And from about 600 million years ago, ever thereafter, green algae are the major primary producers in the continental shelves of the oceans. And the actual microfossil record, which you see in the upper right, actually agrees with that. So that raises an interesting question. We see animals appearing on this time frame, we see oxygen going up on this time frame, and we see photosynthetic algae taking on a new ecological importance. Well, interestingly, if you look at the geologic history of planktonic photosynthetic organisms in the oceans, it mirrors their distribution in space. If you look today, where there are very few nutrients, cyanobacteria still are the dominant primary producers. If there's more nutrients, green algae are important, and only if there's a lot of nutrients do you have things like diatoms. And that's interesting because for most of Earth history, cyanobacteria are ruling the roost. Green algae become important only toward the end of the Proterozoic, around 600 million years ago, and diatoms only later. So perhaps that history of phytoplankton reflects the history of nutrients. Oops. And interestingly, there's reason to believe that the whole geological cycle of phosphate, one of the key nutrients for primary producers, changed on just this time scale. What you're looking at in the upper left there is a whole wall of phosphatic sediments. This is from some rocks that are about 600 million years old in China. And with my Chinese colleagues, we were fortunate to work on this. It has some of the best fossil preservation you'll ever see. But one of the reasons for being interested in this is that at this time period, this one formation has more sedimentary phosphate in it than the entire geologic record up until this time. There's just a big change in phosphate. We think that that has to do with tectonics again, that there's something again on this same time frame, called the Pan-African orogeny in the southern continents. And there's reason to believe that for the first time in Earth history, we have mountains of the size and dimensions of things like the Himalayas today. So they would rain a lot more nutrients into the oceans. That would make for more primary production. That would make for more oxidants, not only more oxygen, but more sulfate. And that would cause greater remineralization of organic matter at depth. That latter is important because if you look at where the phosphate comes for primary production in today's continental shelves, only a few percent of it comes from weathering and erosion. Most of it comes from upwelling of and recycling of materials. So there's reason to believe that, again, for reasons having to do with the gradual cooling of the mantle, that we cross a threshold, and now we have a new tectonic regime that changes the phosphate cycle. That will change oxygen environmental history. And so it may be that tectonics drove changes in nutrients. Those nutrients drove changes in food and oxygen. And that is the context within which we see animals diversify. Okay, well, let me just finish up. The punchlines are, we have a deep geologic record and we can reconstruct our planet's deep history, and both the physical history and the biological history of our planet. And as I said, I hope you've been convinced that I'm not just talking about the early evolution of life. Microbial evolution is the first 80% of the history of life, and we can actually catch glimpses of that history. Also, I hope it's become clear that life and environment have interacted through Earth history. Sometimes major biological transitions are driving environmental changes. Other times, just the reverse is happening. But I don't think we can understand either Earth's environmental history or its biological history without considering the two together. Penultimate slide, just to point out that this isn't just something that happens on the early Earth. We see that over the last 500 million years, through plate tectonics, the continents have continually been moving around. There have been major climate changes. What you're seeing on the right is a portrait of the Earth as it may have looked about 20,000 years ago when major ice age covered much of North America. There's evidence that carbon dioxide levels have changed. There's evidence for short-lived events like meteorite bombardment. What you see here are two dinosaurs on what will be their last date of their lives, looking at this, what looks like a mushroom cloud, but it's really the bombardment of a major meteorite that will change our planet's biology. I'll talk about that this afternoon. And just the final thing that I'll talk about a lot more this afternoon is that we are living in the evolutionary present at a time of unusually rapid change in the Earth system. And so this conversation between Earth and life that has played out over the long time span of our planet's history is particularly relevant to our present and future. So with that, let me just say thanks. I've benefited through time through wonderful teachers and terrific students, great colleagues and sponsors. I thank them all, and I thank you. And with that, I would be glad to try and answer questions. Okay, Natalia, are you on?
Thank you, Dr. Knoll, for that wonderful presentation. I did just want to take a moment to let our webinar viewers know that they are able to submit their questions through the Q&A function on Zoom. And we actually do have one question lined up. Do you have any advice for students interested in making a career out of research?
Yeah, I think the major thing is you have to be curious. nobody that I know, there's no good scientist that I know does research because they want to win awards or they want to do this or that. I think the only thing that can sustain you in a life of research is just to be curious. I mean, I get up, I'm an old guy now, I'm 72 years old, but I still get up every morning just wanting to learn something new about how our planet came to be the way it is. And so, if you're really deeply curious about something, and if you're, that can be very practical, you may want to be curious about how we can cure lung cancer. That's what can drive you. And I think the other thing that I would say is, for many years, the route to success in science has been to focus on something very narrow and sort of drill as deeply as you can into it. But we now live in a world of interdisciplinary research. All the hard problems, climate change, types of cancer, things like this, space exploration, involve integrating disciplines that in previous generations seemed to be separate and whose practitioners didn't talk very much. So I think if you're going to be a researcher, you should learn to do what an old friend of mine, Walter Alvarez, once characterized as the gentle art of scientific trespassing. And that is, don't just worry about what's on your plate, but find out what the person down the hall is doing, because she may have some insights that will help you to understand your problem. And you, in turn, may have something to add to what she's doing. So what that translates into as a practical sense, starting as an undergraduate, is to get as much breadth of science as you can. There's plenty of time to become specialized, but as an undergraduate, even if you're going to be a biologist, you should probably get a little chemistry and physics under your belt, things like that. Even if you're going to be a physicist, you may find that most of the interesting problems are now in the physics of life. So sort of graze broadly, be curious, and then there's a lot of opportunities.
Wonderful. We have another question. You mentioned Mars in one of your final slides. Has there been any compositional research of Martian rocks and soil done by rovers that shows signs of historical microbial activity?
Good question. I had the wonderful privilege of being a member of the science team for the MER rover's Spirit and Opportunity that began their operations in 2004. And what was fun about it is that we could look at Martian sedimentary rocks the same way that we look at terrestrial rocks. And beginning with that mission and with several other, wonderful missions that are still ongoing today, we found that we can look through the physical and chemical characteristics of Martian sedimentary rocks, characterize Martian environmental history. I will say that as of today, there is no unambiguous evidence for life. We now know that there is organic matter preserved in some of these sediments that are several billion years old. I can't rule out that could have had a biological source, but it could be prebiotic chemistry. We know that under many conditions likely to be present on planetary surfaces, we do have organic synthesis. And specifically, there are whole classes of meteorites called carbonaceous chondrites that can have 10% organic matter in them. And that could be a source of some of this organic matter as well. So as of yet, no stromatolites, sadly. Some organic matter. I think it's an open question, but what we are learning is about the history of Mars. And I'll just take a minute to talk about a paper that I published with a very bright young colleague, Robin Wordsworth, last year, which was, if you look at Mars, there's evidence that in its past, Mars was warmer and wetter than it is now. Now it's, hideously cold and dry. I mean, liquid water is needed, stable at the surface on modern Mars. And while that's sometimes been framed as Mars was warm and wet for 600 million years and then things went south, we think it's much more likely that Mars only became warm and wet when there was either massive volcanism or a big meteorite impact, which would put gases like hydrogen into the atmosphere that would be greenhouse gases. They would make Mars warm enough for liquid water, and we have evidence for stream flow and lakes at that time. But rather than being constant for 600 million years, we suggest that it may have been warm and wet for 1 million years and then dry for the next 10 million, then warm and wet. And that produces a very, makes very different predictions for the probability of life. So we don't know yet whether there ever once was life on Mars. There's reason to believe that it wasn't sustained if it ever existed. And the final thought here is that what might be unusual about life on Earth is not simply that it began, but that it's actually persisted for 4 billion years on our planet. That may be unusual, not only in our solar system, but in the universe.
We have one more question.
Sure.
How have mass extinction events impacted the evolution of early organisms?
This afternoon, I'm going to talk about that specifically, but they've had a major influence on evolution.
So why this isn't reaching?
I'm sorry. So yeah. Again, I won't go into it a lot today, right now, because that's going to be the subject of this afternoon's lecture. But I think there's two things to say. One is that mass extinctions have had a strong impact on the course of biological history on this planet. And the other is that despite that, there are sort of vectors in the history of life that run straight through those mass extinctions. So although mass extinctions are important, they're not the only formative agent. There are sort of larger scale features, such as the ones I talked about this morning, where changing environments have made new types of life possible. Had I continued the story upward a little bit, we get land plants, which are basically green algae with a master's degree, coming out on land, changing the land surface in a way that makes it possible to have whole new types of biology on land. So it's a combination of things. And I think the one thing the geologic record shows us is that this is just not 4 billion years of population biology playing out. That in fact, population biology is playing out, but the Earth is changing. It's changing on long time scales, like the history of oxygen. And there are these short-term events that can essentially reset the evolutionary clock fairly quickly.
Okay. What does the geologic record suggest about how global warming will change the atmospheric makeup?
Yeah, another very good question. And again, I'll talk about this more this afternoon, but I think we have strong evidence for atmospheric change over the last 70 or 80 years. There's one of the most important long-running experiments that I think we have is something called the Keeling curve. A man named Ralph David Keeling began in the late 1950s to measure the gas composition of the atmosphere from a station on Mauna Loa in Hawaii every hour. And that continues today. And it turns out that if you look at the amount of CO2 in the atmosphere when the Beatles had their first hit, it's about a third higher now than it was then. And that's an extraordinary rate of change. So because of that, Earth gets warmer because of the greenhouse effect. Also, as you put more CO2 into the atmosphere and oceans, that changes the pH of the oceans, something called ocean acidification. And that makes it harder for things like corals to make skeletons. You also decrease the amount of oxygen in at least the deeper parts of the ocean. And while that's not a big deal, I think in most surface oceans, some of you may know that if you go into the Gulf of Mexico, for example, there are these seasonal expansions of oxygen depleted waters called the dead zone, which is a telling name, which just sort of kills all the animal life living on the floor. There are now dead zones in many different areas, especially the Gulf of Mexico and the Baltic Sea. Those would be strongly exacerbated as global change takes place. So yes, and the environment is changing. That has important geologic consequences. And as I'll talk about this afternoon, extinction in the past provide, I think, a very useful distant mirror on the 21st century.
Okay, we have just a few more questions.
Sure.
You mentioned that the mantle cooled and gave way for the emergence of continents. Is there evidence that the mantle is still cooling, or has it reached an equilibrium?
Another good question. It turns out that when the Earth formed, it was hot. It's hot because there is a heat of accretion as you sort of piled all this stuff together to make the Earth in the first place. And there would have been a much greater abundance of radioactive isotopes than we have today. And as those give way to daughter products, they give off heat. So there's reason to believe that early on, the Earth was hot enough that it basically melted. And that caused heavy things like iron to go into the center of the Earth, giving us the core, and then the mantle and eventually the crust formed around those. Now, the early mantle must have been warmer than today because we know there has been simply a loss of heat from the mantle that continues to today. And the interesting thing is it turns out that when the mantle is hotter, it can't carry much water. And my colleague Rebecca Fisher has done some really nice work suggesting that most of the Earth's water would have been degassed to the atmosphere and Earth's surface early in Earth's history. And then as the plate tectonics starts and as the mantle cools, more of the water that gets subducted back into the interior will stay there. So there's a lot of debate on exactly how much water exists in the Earth's mantle today, but reasonable estimates suggest it's, an ocean's worth of water, perhaps even more. Where you take that ocean's worth of water and put it out on the surface, and it would raise sea level by more than 2 kilometers. So almost all the Earth would be covered in water. And at the same time, when the mantle is warmer, it can't support thick continents the way the present day mantle can. So because of those two things, you wouldn't see much in the way of continental emergence. And that's what the geologic record tells us. And I alluded to that, it wasn't like the Earth was far on until 2.5 billion years ago and then automatically looked like today. It looks like we have this intermediate stage where we do have exposed continents, but probably not much in the way of major mountains. And that then changes only about 600 million years ago. So, one can almost divide Earth history into 3 chapters. which we know are distinguished by different life, different amounts of oxygen. And now that whole thing increasingly seems to be by the evolving nature of Earth's tectonics. So it's the whole system changing in concert.
Great. What would you predict our Earth will look like in 1,000,000 years?
Whoa. I think if you looked at it from the moon, it would look a lot like today. I mean, plate tectonics is operating. Every year the Atlantic is getting wider, but it's getting wider by 2 centimeters a year, something like this. So 1,000,000 years, it won't look wildly different from today. Most of the active mountain belts, in Indonesia, Southeast Asia, etc., will still be operating much the way they do today. I think the bigger questions is, what will the atmosphere and oceans look like? What will the biota look like? I think one thing, and you can decide whether this is good news or not, but we can't keep putting more carbon into the atmosphere because we're going to run out of fossil fuels. Now, there's reason to believe maybe that's a good thing, but it does suggest that on the time scale of 1,000,000 years, the atmosphere might not look wildly different than it will look at the end of this century. To me, the real question, not only on a 1,000,000 year time scale, but on a 100-year time scale, is what will the biota look like? And that's pretty much up to us. I know I can tell you scenarios that will have no more Great Barrier Reef 50 years from now. I can give you other scenarios where people come to their senses and the Great Barrier Reef is sustained. And this goes for pretty much any kind of ecosystem you can think about. So, 1,000,000 years is so far away, and I'll be dead and gone. So I guess I can say anything and not worry about whether I'll be right or not. But I think, the uncertainties on 1,000,000 years are almost smaller than the uncertainties on 100 years, because on 100 years, much depends on us.
Okay, we have just one more question.
Okay.
For people who are looking for life on other planets, would you recommend to put microbes or bacteria that can generate oxygen?
Well, if you're looking for evidence of life that's indigenous to other systems, I think there's sort of 3 ways you can look for life beyond the Earth. In our own solar system, we can go there. We've been to Mars a couple of times. We'll be back again. There's a number of missions, including a really interesting European mission, that are at least going to orbit around some of the icy moons of Jupiter and Saturn, which are, again, interesting candidates for possibly life in oceans beneath a veneer of ice. Beyond our solar system, we can look at nearby solar systems. And our best bet there, this is, I think, where it loops into the question that was asked, would be to see what is the composition of their atmospheres. And again, I think it's an open question whether there is any diagnostically biological atmospheric composition. But I think if we find evidence of oxygen, and in particular oxygen and things like methane in the same atmosphere at the same time, which we have on Earth, that would make people very interested in questions of life. And beyond that, for most of the universe, I think the only way we'll know about life through most of the universe is if they tell us.
Perfect, thank you.
Okay, thank you, Dr. Knoll, so much for everything. Have to wrap up this first portion of the event. We'll see everybody back here at 3 p.m. Pacific time for the technical lecture that Dr. Knoll has continuing. And then Dr. Knoll, we'll be seeing you at lunch very shortly. Thank you again.
Thank you.